Hello everyone, I am Shweta doing first MSc Microbiology. We are going to discuss about physical method of sterilization. First, what is sterilization? Sterilization is the process of killing of microorganisms. Sterilization refers to the process that removes, kills, or deactivates all forms of life, particularly microorganisms such as bacteria, fungi, and unicellular eukaryotic organisms, and other biological agents such as pores present in or on a specific surface, object, or on a fluid. Sterilization was carried out by two methods. First, physical method, and the second, chemical method. Physical method of sterilization has five methods. They are drying, dry heat, moist heat, filtration, and the radiation. The first one is drying. Moisture is essential for the growth of bacteria. 4/5 of the weight of the bacterial cell is due to bacteria. Drying is as a deleterious effect on many bacteria. Spores are unaffected by drying. The second one is dry heat. It has three methods: flaming, incineration, and the hot air oven. The first one is flaming. In flaming, inoculation loops, tips of forceps, and spatulas are held in a Bunsen burner flame till they become red hot. This is the picture of sterilization of inoculation loop by flaming. The second one is incineration. Incineration is an excellent method for safely destroying materials such as contaminated cloth, animal carcasses, and the pathological materials. The equipment used for the incineration process is incinerator. This is the picture of laboratory incinerator. And the third one is hot air oven. Sterilization by hot air oven is the most widely used method for sterilization by dry heat. Hot air oven is used to sterilize the glass bars, forceps, scissors, scalpels, glass syringes, and the swabs. The principle of hot air oven is temperature required for sterilization is inversely proportional to time. During the sterilization of materials in hot air oven, the sterilization time was reduced when the sterilization temperature was increased. The temperature and duration for hot air oven sterilization is 140 degrees Celsius for 1.5 hours, 160 degrees Celsius for 1 hour, 180 degrees Celsius for half an hour. Sterilization by hot air oven kills the microorganisms by denaturation of protein. This is the picture of hot air oven. And the third one is moist heat. It has four methods: pasteurization, boiling, tinderlization, and the autoclaving. The first one is pasteurization. Pasteurization is the fractional sterilization process of milk. Pasteurization technique was first introduced by Louis Pasteur. During the pasteurization, all the spore-forming bacteria and heat-resistant bacteria were killed. Pasteurization was carried out by two methods: flash method and the older method. The temperature required for older method is 63 degrees Celsius for 30 minutes, and the flash method is 72 degrees Celsius for 15 to 20 seconds. This is the milk pasteurization plant in industry. The second one is boiling. In boiling, vegetative bacteria are killed almost immediately at 90 to 100 degrees Celsius, but the spore forming bacteria required prolonged period of boiling. Boiling is not recommended for the sterilization of instruments. It is usually preferred for the cooking the food materials. The third one is tinderlization. Tinderlization is a process dating from the 19th century for the sterilizing substance, usually as food, named after its inventor, scientist John Tyndall. It is still occasionally used. Sterilization can be used to destroy the spores. It is essentially consists of heating the substance to boiling point, which is just a little below boiling point, and holding it there for 15 minutes for 3 days in succession. And the final in moist heat, the autoclave. Autoclave is a pressure chamber. It is used to carry out the sterilization under pressure, which is in moist heat. The mechanism of the autoclave is similar to the pressure cooker. Autoclave works under the principle of Boyle's law. When volume is constant, temperature is directly proportional to time. Temperature and duration for autoclave sterilization is 121 degrees Celsius or 15 lbs for 20 minutes. Sterilization by autoclaving kills the microorganisms by coagulation of proteins. This is the picture of laboratory autoclave. And the fourth one is filtration. Filtration helps to remove the bacteria from heat liable substance such as serum, sugar solutions, or antibiotics used for the preparation of culture medium. The four types of filtrations are commonly used for the sterilization process. They are candle filters, asbestos filters, sintered glass filters, and the last membrane filters. The first one is candle filters. Candle filters are manufactured in different grades of porosity. They have been widely used for the purification of water for drinking and industrial process. They are of two types: unglassed ceramic filters. The example are Chamberlain filter and Dalton filter. And the second one is diatomaceous earth filter. The example are Berkefield filter and Mandler filter. And the second type is asbestos filters. The asbestos filters are disposable and the single-use discs. They have a high absorbing capacity and tend to alkalinize filtered liquids. The examples of asbestos filters are Seeds filters and the Sterimate filters. The third one is sintered glass filter. Sintered glass filters are prepared by heat fusing finely powdered glass particles of graded size. These filters have low absorptive property and it can be easily cleaned, but they are brittle and expensive. And the final one is membrane filter. Membrane filters are made up of cellulose esters or other polymers have largely replaced other types of filters. These filters are routinely used in water purification and analysis of water, sterilization and sterility testing, and for preparation of solutions for the use of parental use. The final method of physical sterilization, which is radiation. In sterilization, there are two types of radiations are used: non-ionizing radiation and ionizing radiation. The first one is non-ionizing radiation. Infrared and ultraviolet rays are the non-ionizing radiation. It is low energy type. Infrared radiation is used for rapid mass sterilization of pre-packed items such as syringes and catheters. Ultraviolet radiation is used for disinfecting enclosed areas such as entryways, operation theaters, and the laboratories. And the second one is ionizing radiation. X-rays, gamma rays, and cosmic rays are the high energy ionizing type. Ionizing radiations are lethal to DNA and other vital constituents. These ionizing radiations have high penetrative power. These radiations are used to sterilize plastics, swabs, catheters, animal feeds, cardboards, oils, greases, fabrics, and the metal foils. Thank you for your patience.